So we left off talking about different types of muscle fibers. And even though there's differences between them, these muscle fibers have more in common with each other than they do with other cell types. Muscle in general has a really unique morphology or shape. When most people hear the word cell, they think of the thing that you learned about in Intro Bio, which is depicted on the left-hand side of the slide. It's essentially a sphere with lots of cytoplasm and organelles floating around everywhere, and there's a big old nucleus somewhere in the center. That's actually not too far off for many of the cells that we talk about in our course, like, for example, an epithelial cell. But muscle looks completely different. As you can see on the right-hand side, myocytes tend to be larger and thicker and more cylindrical than most other cell types inside the body. Since muscle's job is to contract, its only job is to contract, skeletal muscle tries to cram as many myofibrils as possible into the inside of the cell. And that means that the inside of the cell has very little cytoplasmic space in comparison to other cell types inside the body. In fact, most of the organelles inside of muscle, which are represented in this diagram by the mitochondria, tend to get pushed really far off to the periphery of the cell in order to make room for as many myofibrils as possible. Because muscle looks so different, parts of the cell have actually been renamed. So the prefixes myo and sarco both refer to muscle. The cytoplasm of muscle is sometimes called the sarcoplasm, the plasma membrane is sometimes called the sarcolemma, and the endoplasmic reticulum is sometimes called the sarcoplasmic reticulum instead. Now, the reason why myofibrils are so abundant is because myofibrils contain the contractile proteins of the cell. Again, muscle has one job, so these myofibrils are pretty important. The contractile proteins arrange themselves into repeating units that are called sarcomeres, and the sarcomeres are the basic units of muscle contraction. One myofibril contains many sarcomeres. We'll be taking a closer look at the sarcomere when we're together in class, but for now I just want to emphasize that there are really three structural proteins that make up most of the sarcomere. There's a thick filament, which is known as myosin, that's shown in red. There's a thin filament, which is known as actin, that's shown in blue. And there's also a tiny elastic protein called titin, which is actually really difficult to see in these cartoons here. Sarcomeres contract in an oscillatory manner, which is very similar to how a spring or an accordion would contract. Each end essentially moves towards the center. At the top of the slide, you can see an individual sarcomere in the relaxed state and in the contracted state. And I'll point out that the filaments have slid to form the structure in the contracted state. Down here at the bottom, you can see how multiple sarcomeres contracting ultimately changes the overall length of the myofibril. It looks just like an accordion. This is known as the sliding filament model. The filaments refer to the contractile proteins actin and myosin. When this happens on a really large scale, this oscillatory contraction of many myofibrils contracts the entire muscle cell. Now, because myofibrils are everywhere inside of the muscle, sarcomeres are everywhere too. So over on the right-hand side of this image are two cells that have been labeled with fluorescent markers for the thick filament myosin, that's in green, and the thin filament actin, which is in red. And you can see that they're distributed all throughout the cell, but contraction is a really tightly regulated process. And so there's also regulatory proteins that are associated with the sarcomere. If you take a look at the left-hand image, you'll see that it has two regulatory proteins labeled. There's troponin and tropomyosin. Troponin labeled in red, tropomyosin labeled in green. Now, everywhere that you see yellow in this picture is a region of overlap between those two proteins. So from these images, really the big takeaway is that actin and myosin are close to each other, but you also have these regulatory proteins that are present in the exact same spots. So in order to understand how the regulation of contraction actually works, we have to take a look a little bit more closely at the anatomical structure of these proteins that are involved. So up at the top in purple is a representation of the thick filament, myosin. Each individual myosin is shaped sort of like a golf club. It has both a head region and it has a tail region. The tail region is honestly not very interesting to us, but the head region is going to be more important momentarily because myosin heads contain an enzymatic domain that's able to break down or hydrolyze ATP into ADP and phosphate. Down at the bottom is a representation of the thin filament actin. Each individual actin protein is a sphere or a marble-shaped protein. Proteins that have that shape are known as globular proteins. And so when actin is a monomer, it's called G-actin, the G standing for globular. But actin can polymerize to form long chains or filaments. And in this state, it's known as F-actin, the F standing for filamentous. In most cases, when we talk about actin, we're going to be talking about it only in its filamentous form.